Hello, Vinod.
Yes, Jean.
Yeah, so everyone is here. So somebody, students are yet to join, no?
Only 18 students are there.
Okay, oh, yeah, sorry. Yeah, okay. So we'll wait another, yeah. So, okay, I'll talk to someone, okay, no problem. Hello, Salman.
Yes, Gene.
Yeah, so only some 18 students are there. We'll wait for another 5 minutes. So
No issue, no
normally,
issues. Let them come.
so yeah, one more thing. So after your introduction, so we'll give
Hmm.
some time for them to watch the video, or already they have
No,
watched.
video
What
is
is?
already being uploaded, I guess. So there was, is it
No,
already uploaded, right?
yeah, it's already uploaded, but I don't know whether they
Yeah.
have watched or not.
No,
We'll
because
ask them.
that's a long. Yeah, yeah, we can ask them, but that's a long video. They're supposed to watch it. It's not a 2 minute video. It's a 40 minutes video, I
14
guess.
minutes. Okay,
So,
okay.
Yeah.
man, its
So
link is there in chat box.
Yeah, it's
okay,
link is
no
there.
problem. No problem.
So we'll ask them. Otherwise, we'll just go directly to the interaction section. Okay.
No problem. Yes. Okay.
Oh, okay.
Yes. Somebody asked about the quiz. There will be no quiz today. Possibly the quiz will be either on Friday or on a Saturday. We'll ask you people when. I mean, according to your convenience, we'll do that. And also the PPTs. I don't think the PPTs will be shared. So all the materials will be shared with you. All the write-ups will be. All the theoretical part will be shared. But I don't think PPT will be shared with you. So if you see any important point, if you think that is important, please note it down. Otherwise, the PPT won't be shared. What I have a feeling. Yes.
Sir, Vinod sir,
Yes, Harsha.
I just joined right now. Did I miss anything?
No. Only thing is, we clarified about the quiz. Today there is no quiz, but we will announce by the end of today or something.
Okay,
Okay? Yeah.
okay.
So, man, we can start. I think only another 4 5 students are remaining.
Okay, Vinod, we'll start. Is my voice clear? Hello.
Yeah, your voice is clear,
Yeah,
Salman.
my voice is clear. Okay, fine.
So I
Okay.
will. Yeah, okay. Introduce yourself. No problem. Yeah.
So my name is Shomen. I am fourth year PhD student at Center for Neuroscience. I'll be taking your black gold nail staining part, staining in under UB 201 neurobiology. So we'll start. Okay, fine. So today, as a part of UB201 Neurobiology Practicum, we are going to discuss black gold 2 and Nissl staining. I'm going to explain you in detail what basically black gold 2 and Nissl staining mean and what does it do. Before that, I'll give you a brief introduction to what cell types is it gonna stain, or what basically it is gonna show, and what is the procedure. So basically a brief introduction to what we are going to do. So before that, we have different cell types within the brain. Majorly, they are classified into neurons and glia, as you can see here. Just give me a second. Okay, so basically you have different cell types, neurons and glia. Glia can further be classified into macro and microglia. Astrocytes and oligodendrocytes form the macroglia, where the microglia is a different category altogether. All these cells interact with each other and also facilitate their function, and all these cells develop at a different time points during the developmental time scale of an animal. So most of our focus will be on mouse brain. We don't talk about human brain in this experiment. Whatever human brain anatomy and other structures you have studied is all a part of neuroanatomy. Okay, to begin with, all these cell types develop from certain special kind of stem cells that are present in the brain, which are called as neural precursor cells, or NPCs. We are going to call them NPCs from now on. And a mice gestation happens around so basically the gestation period is around 21 days in mouse, that is 3 weeks. So the neurons are the first to develop from these NPCs. The NPCs would give rise to the neurons around embryonic day 14. The peak is around embryonic day 14. The maximum generation of neurons from NPCs is around embryonic day 14. Whereas the development of astrocytes happens around P0, which is basically the peak is around P0. That is postnatal day 0. That is on the day of its birth. The animal is the day when the animal is born. The peak of astrocyte all these process happen, but the only thing is that the peak is different. So the peak of oligodendrocyte generation is maximum around P14, which is postnatal day 14. So all these basically there exists a temporarily overlapping sequence of cell generation. Okay. Now for this part of the practical, we are going to mostly discuss about oligodendrocytes, because these are the major myelinating cells of the central nervous system. In peripheral nervous system, you have Schwann cells. In this case, you have oligodendrocytes. The main difference between oligodendrocytes and Schwann cells is Schwann cells can myelinate only one axon at a time, whereas single oligodendrocytes can myelinate several axons at a single point of time. Interestingly, when they are myelinating, wrapping the axons, they facilitate a faster conduction of electrical impulses in the axon, which is also known as saltatory neuronal conductance. Now, these oligodendrocytes, these myelinating mature oligodendrocytes, they don't directly develop from neural precursor cells, or in short, NPCs. There are different developmental stages as well in between, like oligodendrocytes progenitor cells, pre-oligodendrocytes, and mature oligodendrocytes. These are mature oligodendrocytes, but they are not myelinating. They have to undergo another round of maturation to become mature myelinating oligodendrocytes. Now, these oligodendrocyte progenitor cells, we are going to call them OPCs. You have NPCs, they develop into OPCs, and then they further develop into oligodendrocytes. Now, in an embryonic brain, so basically these OPCs develop from 3 waves. There are 3 waves of OPC generation from NPCs. The first wave happens around E12, embryonic day 12, from the medial ganglionic eminence. This is the embryonic mouse brain, and this is the medial ganglionic eminence. From this, the first wave is generated, and the second wave is of OPC is generated from lateral ganglionic eminence. This happens around embryonic day 15. This first wave happens around embryonic day 12. This wave happens around embryonic day 15. And the last wave of OPC generation, that is oligodendrocyte precursor generation, happens in the cortex, that is on the day of birth, that is postnatal day 0, P0, what we call it as. Okay. Okay. Before that, before I move to methodology, do you have any questions? Anything related to this thing, this oligodendrocytes and other stuffs?
Question is there in chat box.
Questions are there in the chat box? Okay, let me just see.
Hello, sir. I had
Yes.
a doubt. Like, can you
Yes.
go a little bit slowly on this, because many of us didn't have bio, and also even if we did have bio, there's a little bit higher levels.
Okay.
So too many new
Okay.
terms. And
Okay. I'll
one
go.
more thing is, what is myelination? You told.
Myelination. Myelination. I got your question. Is it psi?
Yes, sir. It's I.
Okay. Oh, so myelination is so have you seen the electrical wires, or you have all of you have laptops? You have electrical wires with coated with a rubber, right? Rubber.
Yes, sir.
What is
Sir,
the role?
sir, audio. There's an audio.
Hello.
Sir, there's an audio lag, I believe.
Is it with everybody? Is there a
Yeah,
common
yeah,
audio lag with
Soman,
it?
there is
Yes,
an
sir.
audio.
Yes, sir.
Okay, I'm saying, is it clear now?
Yes, sir.
Okay. It's a simple thing. I'm asking, have you seen the electrical wires in your house? You have a laptop charger as well, where you have electrical wire coated with an insulating rubber. I'm saying, what is the specific role of that electrical rubber coating around your copper electric wire? That's what I'm asking. If you know, give me the answer. Then I'll explain what is myelination. Anybody?
It acts like an insulator
What
so
kind
that
of
we don't
insulation?
get a shock.
You don't get a shock. Okay. 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 Fine. What if you are wearing a rubber glove and then you are touching that copper wire? It's the same process, right?
We still don't get a shock.
We don't
We
still don't
don't.
get a shock. Still don't get a shock. But then you don't need that rubber coating around that copper wire. You can just simply wear a glove, right? Why to waste so much money coating it? Just wear a simple
Because
glove.
all time we can't keep wearing a glove, right? Because the glove is a little bit more
No,
uncomfortable.
there'll be loss of the how do I say this? The
Basically, leakage of signals.
yes, that is what I
Yes,
mean.
correct. So the entire point of having an insulating rubber coating around the wire, naked copper wire, is to prevent the loss of signals, because the electrons will go out in that case. Now, in this case, in neurons, the electric impulses are not in form of electrons. It's not a ion. It's basically ionic in this case, sodium, chloride, and all those stuffs. But in this case, when you are coating, so this oligodendrocyte cells, they form a coating which is formed of lipid, which is again insulating, equally insulating as rubber. So then they coat it around the axon, naked axons, and when they do so, it prevents the signal leakage. Also, they don't make a continuous myelinating stretch. If you can see this image in this presentation, you can see that there is a myelin wrapping around the axon in here, and there is a very tiny gap in between, and there is the next myelinating myelin wrap around on the axon. Now, why is this the case? So, first of all those ionic currents that flow, this because that ionic currents that are generated is generated by the formation of a gradient across the two extracellular as well as cellular space. Now, all those channels, sodium or chloride channels, are concentrated in this region. Because the signal leakage cannot happen in this region. So whatever exchange happens in this region, which is also called as node of Ranvier. This space where there is no myelin is called as node of Ranvier. Now, so since the exchange is happening only in these nodes of Ranviers, the conduction of electrical impulses is fast, and it follows a kind of a jumping kind of a motion. Therefore, it is called as a saltatory conduction. The entire point of having a myelination and myelinating the axons of the neurons is simple: just to have a thick insulating coating which will prevent any signal loss to the outside from the axons. That's all. This is myelination. Okay. What is P and E? P is your
Sir,
postnatal day. Yeah.
it's okay, sir. You can continue. I'll take the question next. I'll ask the next question.
Okay. Then so P is your postnatal, that is after birth. E is your embryonic. What is this? What promotes myelination? NE. What is that? TF. Transcription. Where is it? I mean, what is the?
Transcription factor.
No. What is the question then?
Like what promotes myelination? Like you had shown in one of your
Okay.
slides, right? Different stages.
Yes. Okay, what promotes? There is a lot of there are a lot of factors, not only transcription factor. Transcription factor is definitely important, but it also depends on how the neurons are working and what factors are being secreted by astrocytes. So I didn't add the slide, but I'll just give you a brief idea of what is happening in here. People are trying to find out what happens when there are sufficient number of neurons formed from NPCs. Then these neurons instruct these NPCs to form astrocytes. And when sufficient number of astrocytes are formed, then the NPCs are instructed to form oligodendrocytes. But interestingly, nobody knows for sure whether it is the neurons instructing the NPCs or whether it is the astrocytes instructing the NPCs to form oligodendrocytes. Now, there are many transcription factors, as you have mentioned, many transcription factors which are involved in the formation of oligodendrocytes from NPCs, including OLIG2, OLIG1, SOX10. There are many. That's a lot of, I mean, it will be a lot in detail, but don't worry. You can always go back and discuss about these stuffs, but that won't be a part of this practical. Don't worry about that. Okay. Another doubt is, if NPC starts to give rise to murine neurons before it generates glial cells, how does an initially formed neuron derive nourishment and excrete metabolic waste without the aid of glial cells? Okay, this is interesting. I'll tell you something. When the neurons are generated, they are developed. So by the way, neurons, once they develop, they're postmitotic, and in most of the regions in the brain, except some regions like dentate gyrus or other regions, they do not form again. And the second thing is astrocytes are generated later. There are other cell types like neurons. They don't generate a lot of waste. That will taken care of. There are other cells like microglia and other stuffs. They will take care of it. And other than that, when the animal is born or the animal is around, I mean, if you see clearly, the development of astrocyte is already happening from E12.
Sir, you are not audible.
Yes, sir. You're not audible, sir.
I'm not audible?
No, sir.
Okay, okay. Am I audible now?
Sir, your
Yes,
voice
yes,
is
Yes,
we can't hear you.
yeah,
talking.
sometimes your voice is breaking. I think
Yes, sir. There's a lag.
yeah, sometimes breaking. Can you check with the connection or something, or maybe headphone? Or you're using headphone or something?
Yeah, I'm using a headphone, but okay, just give me a second. Just give me.
In between some seconds only, it happened like that.
Oh.
Just give me a second. Yeah. Is it fine now?
I think yes. I think yes. I think yes. I think yes.
Sir,
Now
there's echo.
Now it's an echo.
Is it fine now?
Yeah, yeah. Okay, you just continue. We'll see like that.
Yeah,
Now,
I'll continue. Yeah. So, okay. Yes. So, okay. The next. Okay. Let me explain the previous question. So, all the cells are almost developing simultaneously. If you see, there are some astrocytes being developing around E14 as well. And there are also oligodendrocyte generation happening around E12, as I have mentioned, from the first wave OPCs. Now, interestingly, all those neurons that are formed are more or less immature. They have to reach their destination. Though they are generated, they have to reach their destination through radial glia. Once they reach their destination, they start to function. That takes some time. And if there are waste generated, there are certain cells like microglia which are not generated from NPCs. They will also take care of it. They are also involved in synaptic pruning as well as pruning of myelin as well. So yes, this all process is a very coordinated process. Only thing that I'm talking here is that the peak of neuron generation happens around E14. Similarly, peak of astrocyte happens at a later time point, and oligodendrocyte happened around P14. Is it audible now?
yes, sir. I have
Okay.
one related question. Are the microglia derived from blood cells?
No, they are derived from your basically no, not blood cells, but they are derived from what you call as mesoderm. So your macrophages have the similar origin. Similarly, microglia have the origin from the mesoderm itself. So all your macrophages that you see in peripheral circulation is you basically which are phagocytic cells. Similarly, microglia in the brain are phagocytic cells. Their major function is to phagocytose and clean the waste, but they also maintain a lot of other functions as well.
Okay, sir.
Okay, so he says, unlike our computer charger wire, why they are unmyelinated. That's what I'm saying. Why there is a regions of gap? There are gaps in between. These gaps are called nodes of Van Leer, and these gaps are the ones because if the neuron axon wants to transfer the electrical impulses, this is an ionic current. It has to maintain a gradient across the membrane, right? And to maintain that gradient, it needs the transfer of ions across the membrane. But where there is myelin cover, it cannot do that with the external environment. It has to do it in these regions. So these are the regions where the channels, the sodium channels, chloride channels are most concentrated, and then they can exchange the ions in these regions, and as a result, the conduction is proper. Just this actually increases the what you call the speed of conduction, which also is known as a what you call as a saltatory mode of conduction. If you go and read about this, you'll get a very nice 2-3 liners on this. Okay, other than this, any other questions?
Someone just 1 minute. Yeah. Hello.
Yes, tell me.
Yeah, this most of them they have not read anything about this neuronal conduction and all. Say I
Okay.
think the browser theory browser next this week maybe will go through this one. So
I
they don't
think
know anything. Yeah, even the third practical it is going to they are going to use the neuron software. Then they
yes.
will explain how
So
exactly this
yes,
neuronal conduction is happening also.
yes. So
So
basically, in your next practical, these people will be explaining you all these neural conduction because I think they will take care of EEG and other stuffs as well. So
yeah.
yes, so for now you can just know this, and the next practical they will explain you all the other technical details of it. But for now you just understand this that this myelination is absolutely important to maintain a faster mode of conduction over long distances. For short distance, many of the neurons, not all the neurons are myelinated. Many of the neurons are not myelinated at all because there are smaller neurons and they don't need faster conductance over long distances. Only those neurons which require faster conductance over long distances, they do all these myelination stuff. Okay. No, Avikalpa have asked a question that won't neurons most likely be the ones responsible for the formation of oligodendrocytes, as neuron mostly are the ones that are responsible for the formation of. It's not responsible. They are not responsible formation. They're just instructing that there are sufficient neurons. Please go ahead and form more astrocytes now. So it's not forming them. It's like instructing the NPCs, rest of the NPCs, that now you don't need to form more neurons. Now we are enough. You go ahead and form astrocytes. So, but we don't know what is instructing the NPCs to form oligodendrocytes. That's not responsible. They are not responsible. They definitely provide other stuffs like glucose, provide signaling and other stuff. But we don't know yet that it is whether the astrocytes, and it is possible that they both are coordinating with each other, that is astrocytes and neurons, to form the oligodendrocytes. Okay, other than this, there is exchange of ion across membrane while electrical signal conduction occur in neurons.
That's a
Okay,
explanation, so man, that's noted out. So
okay. Oh, it's your Manish. Okay. Oh, one more thing. If you are really interested in knowing how the conductance happen and what is the physical basis of it, please go and read about cable theory. If you read about cable theory, you will be able to know in a very simple terms what exactly regulates the neuronal conductance. You don't have to know a lot of stuff. It's just very simple theory which explains how the conductance is regulated on a very physical terms. Okay, should I move ahead? Okay, I'll move. So before I explain what the staining procedure is and what does it do, I'll explain you briefly what the methodology is. So first, most of the stainings require perfused brains. So for this, we are going to do annual perfusion with 4% VFA. This will be followed by brain dissection, and then it is followed by post-fixation and sucrose treatment. This is followed by brain suctioning using a cryostat, which maintains a temperature of minus 18 degrees Celsius to minus 20 degrees Celsius. And this is followed by staining and mounting. And once we are done with the staining, we observe it under the microscope. Now, before okay, now for animal perfusion, before I explain what animal perfusion means, you all should know what double circulation is. So anybody has any idea what double circulation is? This is a part of your class 10th and 12th syllabus. Anybody? No, nobody knows double circulation? Interesting.
So it's basically a mode of circulation where there are two channels. One carries the purified blood and other with carbon dioxide. I mean,
Okay.
the
Okay. Anybody else?
I think it's basically, I mean, it's called double because it like has two systems, like two circuits, you can say. So, like one of them basically takes blood to and from the lungs, and one of them takes blood to and from the tissues. So,
Okay. Correct. You can take it either way, because since there are two circuits available, you call it as double circulation. Or if you want to remember, simply it passes through a single body or organ twice. Double circulation. I mean, the way it depends completely upon you. But majorly, why it is called a double circulation? Because there are two circuits involved, two circuits of circulation involved. One is a pulmonary circuit, and the other one is a systemic circuit. Now, what happens? From the different body organs, the deoxygenated, the blue ones represent deoxygenated blood with devoid of oxygen, and the red ones represent oxygenated blood, which is represented in red here. And from the body organs, through vena cava, which is a major vein. Vena cava is a very big vein. It carries all the deoxygenated blood to your which side, right or left? Okay, no response. So this is your right side of the heart, and this is the left side of the left side of the heart, mouse heart. So the deoxygenated blood goes into the right side of the heart. This region is called right atrium. This region is called right ventricle. So from right atrium, it goes into the right ventricle, the deoxygenated blood, which doesn't have any oxygen. And this region, this valve, which is guarding the opening, is called as a tricuspid valve. Tricuspid, Tricuspid, tricuspid valve. From this, it reaches the ventricle, the right ventricle, and then from there, through the pulmonary artery, it goes to lungs for oxygenation. When it reaches the lung, it takes up the oxygen, the blood takes up the oxygen, and then now it has become the oxygenated blood, which is depicted in red. Then through pulmonary vein, so arteries carry oxygenated blood, but pulmonary artery carry deoxygenated blood. Similarly, veins carry deoxygenated blood, but pulmonary vein carries oxygenated blood. This is the opposite. And now the oxygenated blood reaches the left atrium. This is the region, and through the bicuspid valve, it goes into the left. From the left ventricle, the oxygenated blood is again transferred to the same body or throughout the body or the organ from which the deoxygenated blood came. Now, this is very important. Remember this double circulation; it will come handy throughout your life. And now, what we do, we take the animal, anesthetize the animal, whatever is being suggested by your ethics committee, you use that same anesthesia and anesthetize the animal and expose the thoracic cavity. So basically this chest region where the heart is called the thoracic cavity. Expose it, and then you have a needle which is going to supply your 1x PBS. So first what we do, we perfuse the animal with saline to remove all the blood. Once all the blood is removed, what you do, you inject PFA, 4% PFA. I'll tell you what is the role of PFA. So you insert that needle into the left ventricle, which is this region, left ventricle, and then you start injecting whatever solution you have slowly, gradually. You have to be very slow while doing so, and simultaneously make a cut at the right atrium, this region. So basically what will happen, whatever solution is going in, it will circulate throughout the entire body and come out of this. So once you are injecting saline, you will be injecting the saline from this place, and it will pass through the entire body and come out of this. So first you pass the saline to remove all the blood, and saline will be isotonic. Make sure it is isotonic, pH 7.4, and it should pass through the atrium. And then once the entire blood is removed, then you pass 4% PFA. Now, what is the role of 4% PFA in here? The 4% PFA is basically paraformaldehyde. What it does, it cross-links all the proteins that are present within the cell. So it's basically a snapshot of that stage. You get a snapshot of that stage. So everything becomes fixed. So nothing, no other activity, no metabolic processes, no changes happen in the cell once you perfuse them with 4% PFA. After this, once the animal is perfused, you have to dissect the brain out. What you do, expose the skull, remove the skull, and this is the brain that you have. This is how it should look. You take the brain out. This is the dorsal view of the brain, and this is the ventral view of the brain. So you have to take the brain in its entirety. Once you have taken this, once you took out the brain, you have to transfer the brain to 4% PFA for 16 hours, that is overnight incubation. Okay, how do you remove the skull? Using your dissection instruments, you can cut the skull. You just make, okay, fine. So what you do, you make an incision here, you make another incision here, and then you make incisions, what do you call, parallelly to the first two incisions. And then you remove the skull with your forceps, strong forceps you have to take, and then you have to remove the brain out. Now you keep it in 4% PFA for overnight post fixation. Once you have the next morning, the first thing in the morning you have to do, you have to remove the PFA and then wash the brain with 1x PBS, that is saline basically in this case, and then you transfer it to 30% sucrose. So 30% sucrose, initially the brain will float on the surface, but you have to let the brain sink completely, which will take about 2 days. And what is the role of sucrose, by the way? It acts as a cryoprotectant because we are going to section the brain at -20. It's called cryosectioning. And just to prevent the formation of water crystals, ice crystals, which might damage the cellular architecture, cause mechanical damage. So you treat the brain with sucrose, which acts as a cryoprotectant in this case. Any questions in here? Nothing? Okay, I'll move ahead then. Okay, then this is a cryostat. Once you are done with, once the brain completely sinks in the sucrose solution, you take the brain out, you freeze it accordingly. I mean, different labs use different freezing techniques. You can either use isopentane maintained at -80°C, or you can use certain embedding solutions as well. So once your freezing is done, you use this machine to um, uh, prepare thin brain slices. It can be 30 micrometer, it can be 50 micrometer, depending upon what is your purpose. Whatever experiment you want to do with the sections, depending upon that, you decide the thickness of the sections. Now, you can either do an axial section. In humans, this is the plane, and this is how a mouse axial brain slice looks like. This is a coronal section, and this is how a sagittal brain slice would look. So in your case, we have all done all the three. Then once you are done with your sectioning, now you have to proceed with black gold 2 staining. Now black gold 2 staining is a very interesting technique. Initially they had black gold 1, so they have developed upon the black gold 2. So interestingly, just much like Golgi staining, which is basically used to stain neurons, nobody knows exactly how this black gold 2 stains the myelin. So basically it stains the myelin. It's a gold based stain. It has complexed gold in it. So it provides, this black gold 2 version provides much better resolution, contrast, and also it takes a very short, the staining entire procedure is for a very short duration as well. And during this experiment, we also use sodium thiosulfate solution to inhibit the staining. So once we get optimal staining, we stop the reaction using sodium thiosulfate, which not only removes the excess, but it also stops the reaction. It inhibits the reaction. The protocol, I have already given the link in the chat. And when the black gold 2 staining is done, we counter stain with crystal violet, which is also called as Nissl stain. It stains all the Nissl bodies in the cell, basically Can you tell me which cell among neurons, astrocytes, and oligodendrocytes have the Nissl bodies? Anybody knows that? First of all, Nissl bodies. Do you know what are Nissl bodies? You must have studied in your class 9th.
Proteins.
Basically, these are ribosomes, free ribosomes, and the ribosomes attached with your RER, rough endoplasmic reticulum, in the cell. So these are ribosomes. Remember this. So Nissl bodies are ribosomes. Since these were discovered by a person named Nissl during his experiments, so he named it as Nissl, and he was working with this stain crystal. So it gives you a very brilliant bluish color, and it basically these are also present in astrocytes as well as initially the person who discovered the stain, he thought that this only stains the neurons, but later it was found it also stains astrocytes and other cell types as well. So what we are going to do, we are going to do black gold 2 staining, which is going to label all the myelinating fibers in the brain tissue sample, and then once it is done, we are going to counter stain, as you can see in the video, with crystal violet, which is a Nissl stain, and it's going to label all the cell bodies. It will label all the cell bodies of the cell, in the brain. Okay. After that, this is a previous previous images. So this is how a transverse section would look like, stained with all these maroon. So maroon regions, the reddish regions are your myelinated fibers, and these bluish ones are what you call as the Nissl staining. This is a coronal section, and this is similarly a sagittal section. Okay. Why are we removing the blood before removing the brain? Okay. First of all, if you don't remove the blood, you'll have all the blood cells also fixed. You don't want that. If the blood cells gets fixed, it will give you a lot of background. So the signal to noise ratio will be too high. So you won't be, sorry, not too high, it will be too low. So it will be very difficult for you to tell which is your desired signal, which is not your desired signal. So you have to remove all the unnecessary autofluorescent or auto coloring stuffs from your body. And once you remove the blood cells, so then you'll have you can always, the signal to noise ratio will always be better. You can always tell with surety, complete 100% confidence that this is your signal. Can we use glycerol as? Yes, some people do use glycerol as a cryopreservant. In this case, we are using sucrose because it's easier to clean. Sucrose is easier to clean, and also it's cheaper than glycerol. But it's also good because it's anybody knows how sucrose works? Just think about it. Everybody knows it. How sucrose might be replacing water molecules or removing the water molecules. Anybody? Just think about it. How does sucrose removes the water molecules? Have you ever heard about osmosis? Nobody heard about osmosis.
Okay, so I think it happens by if you put sucrose, the outside solution will become
Hypertonic.
hypotonic, and
Hypertonic.
so water comes out, and
Yes.
the inside will reduce. The water
Yes.
inside will reduce because all the water comes out. It's like putting
Yes.
salt and getting the water out for preservation,
Oh,
something like that.
yes. So you remove the water molecule in that manner. Okay, there is one question which says, okay, first of all, what is black gold 1? Black gold 1, so the company which makes black gold solutions, they made a earlier variant which is called black gold 1, and they later improved that product to make and named it as black gold 2. It's just the improved version of black gold 1, nothing else. Another thing is, what is signal to noise ratio? Okay, let's say I'll give you another image, show it to you here. You see this is a myelin stain. This is a corpus callosum in this case, and this is a cortex. So in this case, this reddish background that you see is myelinated fibers. So in case if you don't remove the blood, so what blood will pick up everything in a very non-specific manner. So you won't be able to tell whether this is the corpus callosum, whether this is the cortex. Everything will be stained almost in a similar manner. So the background staining will increase. So your signal is this, and whatever you get other than this is a noise. Okay. So this is a magnified version of whatever images that we had. So this region represents the cortex. This patchy region represents the striatum. This is corpus callosum, which is located, which connects the two hemispheres. And this is cerebellum, which helps in the coordination of voluntary movements. And this is your hippocampus, which basically most majorly associated with your recent memory recall. There are other regions as well, but this is the major region which is more associated with your recent memory recall. Any other questions? Don't worry, whatever your conduction, nerve impulses, your cable theory will be taken care of in the next week. But it's just the beyond the scope of this practical. I mean, we can discuss offline, but anything related to this? Anything, any questions? No more questions? Nothing, right? Okay, good luck for exams. So your exams will be mostly on Friday or Saturday. We'll discuss about that. Okay, why are we using isotonic solution in the starting? Why are we using isotonic solution? So when are we using so okay, let me go back to the perfusion step. Okay, this is the perfusion step. So when you are injecting an isotonic solution, why are we doing that? Just to maintain the osmoregulation. I mean, basically so if you're giving a hypotonic or a hypertonic solution, the cells will lose water and they will start shrinking or there will be damage to the cellular architecture. So before you add PFA, everything should be isotonic. Before you cross-link all the proteins and take a snapshot of that stage, everything should be isotonic. Otherwise, there will be a lot of cell damage. The cells will shrink. Or depending upon whether it's a hypotonic or if it is a hypotonic solution, the cells will swell and they will burst. So all the architecture will be gone. So if you want all the cells to be intact, you have to first use an isotonic solution. An isotonic solution is a solution where there is no movement of water molecules across the membrane. So once you are done, use the isotonic solution to remove all the blood on the blood cells. Then you inject your PFA to fix and cross-link all the proteins. Okay, there's another question. Why can't the slides be shared? Because this will be used for the next year as well. And yes, it's not, I mean, we cannot share these because of the property of ISC as well. But don't worry, every handout will be given to you. Everything which you need to know will be given to you. So not to worry about that. And the exams will be exactly whatever I have explained, whatever the previous whatever has been explained in the previous practice, only that thing will be there. So just note down whatever key points, even if this is not in the slide, I might be mentioning small things here and there. Just note down those. If you do have doubt, come back again, ask me, don't worry. So before the exams, even if you can contact me, no problem. But yes, slides won't be shared. Don't worry. That's all. Anything else? So if you want, I mean, I'll share all the details. I'll pass on a handout. I'll ask Vinod to mail you all of you to that PDF, to pass that PDF, and everything will be mentioned in that. Just go through it once. It's just 10 minutes or 15 minutes handout. Once you are done with that, you can always give that exam. An exam won't be tough. So it will be a multiple choice quiz with multiple answers correct. There will be 30 questions, each one of them carrying 1 mark each. Okay, can you take?
Yeah, summary is completely from the first experiment and second experiment, no? Just we have planned like
both combined. Both combined.
So yeah. And about the assignments, we need to give them a question, you know, not just
assignment. We can, once they are done with the quiz, we'll give them the report, and once we give them the report, we'll give them the weekend, or Saturday or Sunday. They can always submit, come back on Monday. We'll decide that. That's not a problem. Okay. First, they have to go through the quiz, and also somebody's asking, can you recommend any book for this course? Okay, I gave you a protocol. There are research papers for Black Gold too, but I would say don't go through the research papers because they don't explain a lot about how it is working, because nobody knows it. And about in general, if you want to study neuroscience, I would suggest there's a book called Fundamentals of Neuroscience. You read that book by Kendall. You read that book, you'll get a lot of idea about how even the molecular part as well as the system part, you'll get an idea about how the brain works. So that's a Bible for our neuroscience. Is it Does it clear? I mean, does it answer your
Sir,
question?
what book is it? Can you repeat again?
It's a Fundamentals of Neuroscience. Fundamentals of Neuroscience by Eric Kandel. Read that book. It will cover most of your theoretical part. Anything else? Nothing
So
else,
the
right?
slides part of
Yeah.
it, is it a policy of the neuroscience department, or like other bio courses, they did share the slides, that's why.
I don't know what they are doing, but I have a feeling I won't be able to share the PPT. The simple reason is that because we are going to use it for the next year as well. They might change the practical, but for us, for the neuroscience, I don't know what other department for the neuroscience.
Sir, but how does
This
it affect
practical
the
stays.
next year's
Yeah.
batch? Sir, how does
What
it
is that?
affect the next year's batch? Like
See,
the slides.
do you have any problem with me sharing the PDF with everything in it? No, right? If you have
No.
problem, just tell me. No, right? Then I'll say share the
No,
same
it's
thing.
like more colorful. The slides are more
No,
colorful.
don't okay, don't worry. Everything will be shared. I'll give you a very interactive PDF. Don't worry about that. It will have more details than the itself. It will be in more detail than you have it in here. So don't worry about that. Will be shared. Don't worry. Your exam,
Sure.
it's just that everything, whatever is there in the exam quiz, you will get everything there. You'll get more than what is there in the exam. But just
Sure.
go through that once. Once it is shared, go through that once. It will take maximum half an hour, and then you can always be able to sit for the exam. Don't worry. Anything
Sure.
else?
Thank you.
Yeah, thanks, Salman. I think they don't have any questions. Okay, just we can
Okay.
wind up. We can wind up the session, or what do you
Okay,
think?
we can, if they are okay with it, we can wind up the section. Also, I'll yeah.
Hey, video they have to look and they have to come for the any question answer section, right?
So already they
Okay.
have watched the video. So I think it's a
Did-
15 minutes
Did
video.
any? I think they have watched the video. When did
They
you
have
upload,
watched.
Vinod?
Actually, we uploaded, but we shared the link today itself.
Oh,
Okay. So nobody, I mean, is there any, I mean, have you watched the video, or you want to go back and watch and come back? Nobody's saying anything. So
yeah,
not
nobody's
watch.
They are
They say
in chat
not,
box. They are mentioning that they
not
didn't
watch,
watch.
not watch.
not
So
watched.
if you want to go back and watch the video for 15 minutes, you please do that and come back and ask me questions. No problem. Okay, you please go back and watch it. The link is there, and I'll be here after 15 minutes. Exactly after 15 minutes, I'll be here. So
Yeah.
we meet
Yeah.
around 3:05 then.
Oh, yeah. So we know for this one, the entire batch is just upload this video. I think they can watch and otherwise again in
Yeah,
between the
we know
practical.
upload the video for the entire batch so that the next batch can come and it's a 15-minute video, right? It'll take some time. Some people will pause it and yeah. So you just upload the video, no problem.
It's not like that because
Okay.
in this batch around 10 people are watching and another 15 people are not watching and coming back again. Those who are watching previously, their times also wasting. So I thought it's better to be upload today, then everyone will watch at the same time, then
Okay, no problem.
nobody's
Okay.
time is wasting, right?
No. Yeah, what we are saying, like that you upload the video, okay? Then we can ask them like that you just they will get enough time, no, just to watch.
Just mention. I mean, just mention one thing that please pre-watch the video. If you are not watching the video, we won't be able to explain the queries that you have. Just simple.
Yeah.
Just upload the video to common because in 15 minutes video, I don't know how many will come back to discuss. So just upload the video. For this batch, it is okay. We'll just discuss this. I'll come back after 15 minutes. No problem.
Yeah.
Is it okay with you, Vinod? That's fine then. Otherwise.
Yeah. Okay.
Okay. Another thing, I'll what do you call? I'll give you the handout. I'll give you the handout by today, and you just share it with them and let them everybody read by Friday or Saturday whenever the exam is, and then they can always come back for the exam. Can we meet at 3:10? Okay, no problem. We'll meet at 3:10.
Hello?
Are you done? Are you people
Yeah.
done?
I had a question.
Yes.
So why are you not like covering the black gold stain with the gold foil and like keeping it away from light?
Why are we keeping it in foil, or why aren't we keeping it in foil? What is the question?
Why are we keeping it in foil?
Because it's
And like
light
covering?
sensitive. It is light sensitive. Over time, if you keep it in light, it will degrade, it will break down. So it won't work. That's the one point why it's a light sensitive stain.
Can you explain what step by step alcohol cleaning does?
Okay, what does it do? Okay, ethanol. Okay, so this so you what you have to do, you have to pass the once you have done with the staining, both nickel as well as black gold, you have to pass it through different gradations of alcohol in a sequential manner. So let's say you have 50, 70, then maybe two gradations of 90, and then 100. So what does it do? It has to replace the water that is there in your sample. So this is basically a dehydration step where you sequentially remove the gradually remove the water molecules from what you call the tissue samples that you have stained. So during this processing, whatever water has been absorbed by these sections, you have to gradually remove it through these gradations. Why don't we do directly at 100? Because then there will be a mechanical damage to the sample itself. It will be a shock to them. So generally, you put it in a 50% where the 50% alcohol, 50% water, and then you go to 70% alcohol, and then you go to 95, and then you go to 100. So basically, it removes whatever the water molecules are there in the process sample, and then in turn it gives you a better resolution as well. And the xylene step is majorly for your to remove. It's basically a defat agent. Basically, it removes all the fat or the lipid molecules that are, I mean, not lipid molecules, but it clears the tissue. It clears the tissue, and as a result, it becomes when you observe it under the microscope, the resolution is a better. Anything else? Also, it resolves dpx. So the dpx that you use as a mountant later, it dissolves your it is dissolved, it can be dissolved in xylene. So when you have the xylene on the slide, it's easy to adjust the cover slip with dpx. Anything else?
Yeah. Why does the black gold stain cost so much?
It has. Where did you see the cost? The link?
Oh, it was. Yeah.
It costs
No,
because I mean, yeah. So
so
first of all,
I think it was in the video.
Yeah. So okay, yeah. So the kit was there, right? So generally, first of all, this is not synthesized in India. So whatever you get, you import it from US or the wherever the company is, the Merck Millipore. Second, you have to pay a lot of import duty there. And the other thing is that it has complex gold. And believe me when I say that whatever biotechnology products are there, even if the manufacturing cost is like 5 rupees or 10 rupees, it will cost you thousands if you buy it from a company outside. So let's say there is something called distilled water, right? If you have a distilled water machine, millipore, millicube machine, you don't even care about the water. But if you buy it from outside, it will cost you maybe some 1 ml of water. They give you 1 ml of water for almost 5,000 rupees. So that's all. I mean, everything that you buy in biotechnology is actually costing, I mean, very not very cheap, I would say. Anything else? Any more questions?
Yeah, so with respect to the gradations of alcohol, so why exactly are we using various different gradations? Why can't we directly go to the last one?
So you missed that step when I said that it gives it's kind of a shock. If you go directly to 100% alcohol, it's kind of a shock to the tissue itself. It's like instant dehydration. So if you do instant dehydration, there will be mechanical damage to the sample itself. So you go gradually, slow by slow, step by step, to remove the water molecules slowly. Yeah. What is the other question?
Yeah, and with a follow up on this, so when you're saying it causes shock, so how do you know between any 2 points that we have taken, it doesn't cause shock?
Okay. There's another point to it. Not only it's dehydrating the sections, It's improving improving your visualization as well. And another thing is that it also removes the excess color. So when you have done the Nissl staining, we are done with crystal violet staining and Nissl staining. If you do it practically, you'll observe that while you're passing through 50%, 70%, 95%, it is the excess stain, which is the background stain, will remove, the section tissue sample will lose the color slowly. So you have to time it in such a manner that you don't lose the desired signal, but instead the background staining is lost. Also, your another question was what was the other question, Rashi?
The follow-up one.
Yeah, follow-up one.
Yeah, so basically I was asking, like, when you're mentioning that you're not giving it shock, so how do you know that, at whatever concentrations you have taken it is not going to give a shock? Maybe something in the transient phase of the two also can cause shock, right?
Okay, anyways, I'll tell you something. When you do perfusion, right? When you do perfusion and the sucrose thing, cryoprotein thing, this generally the size, the brain will shrink actually in general, but there will be no overall gross changes or deformities in the cellular architecture. Similarly, when you do dehydration, there will be some shrinkage of the tissue sample as well, but there will be no gross deformities. So when you do gradually, these are all standardized procedures. Previous people have standardized, even some labs standardize, even some labs use 70% directly and then do 90% and then 100. In my lab, what we do 50%, 70%, and then twice 95%, and then 100% twice, and then xylene. So depending upon what purpose it is, what staining it is, and how thick the sections are and what plane it has been sectioned in, you change the protocol accordingly. It is standardized. It doesn't give a lot of shock. I mean, shock in the sense it shrinks, but it doesn't cause any cellular damage, architecture damage. But while if you go directly to 100%, you can do this cool experiment. You can clearly see the sections will fall off the what you call. So they are mounted on the slide, right? When you pass it through the gradation directly, when you pass it on to 100%, the sections will fall off because they will shrink, they will crush, and they'll fold from the edges and all those stuff. So yes, you don't do that. It's all standardized procedures. You have to gradually remove the water molecules from the sample, and that's all. Does that answer your question?
Yeah. And then the next thing was we are like kind of making a barrier by putting nail polish, etc. Why didn't we use a cavity slide?
Cavity slide. Okay, that is first of all, a cavity slide will cost a lot. Second thing, this is an easier way to do it. In general, in my lab, what we do, we don't use a nail polish. We use a hydrophobic pen. It's a pen which when you mark the boundaries, it actually doesn't allow the water to flow through it. And in this case, we are using nail polish. That's just a very economical way of doing the experiment. You can use a cavity slide as well, but the cavity slide will cost a lot more than this. Say a Blue Star slide box of 70 pack of 72 will cost you some 90 rupees, whereas similarly a cavity block slide will cost you around, on one pack will cost you around 2,000 to 3,000. So definitely this is economical, so use it. One nail polish will cost you around 10 to 20 rupees. That's all. No other reason.
Oh, and the next thing is the sodium thiosulfate. Can you explain its role?
Yeah, it removes all the excess. First of all, two role. It removes the excess black gold, cleans the slide. Second thing, it inhibits the reaction. I exactly don't know what chemical reaction is going on in here, because this is also patented. Black gold 2 is patented, though the many people have tried to see how black gold 2 stains, but more or less they say it stains for the myelin proteins, but which myelin protein is being stained, it's not clear even through the experiments. Yes, sodium thiosulfate, it inhibits the reaction, black gold 2 reaction, which stains basically the myelinated fibers. The major role is to inhibit the reaction and clear off excess stain. That's all.
And DPX is used for?
DPX is used to mount. So once you have processed the slide through xylene, you add DPX to the sections. The simple role of DPX is to provide a better refractive index, so that your the imaging is proper. When you want to look under the microscope, it gives you a refractive index of around 1.58. So it's the best for your what you call visualization. And the second thing is it's freely miscible in xylene. So when you have xylene treated sections with traces of xylene in it, and you add DPX and you put the cover slip, there is a possibility that you might get a bubble or the cover slip is not properly placed. In that case, it gives you a window where you can adjust the cover slip accordingly. That's all. Two roles: freely miscible with xylene. Second thing is it gives you a refractive index of 1.58 for better resolution.
So there was also this oil that we could use, right? The lens oil. Won't that work?
No, that is for higher objectives, right? When you need oil, why you want to put oil? Because if you want to do 40x, 100x, depending upon what microscope, what objective you use oil. But why you want to put oil every time on your slide? Because you have to clean it every time you want to keep it, store it once you're done with the imaging. You do only, so whenever you use oil for your imaging, you start with a lower objective, 20x or 10x or 40x, and then you move to a higher when you know that after this you're going to stop the experiment. But if you want to move to a lower objective again, why you want to add oil? If your purpose is solved with this objective that is 10 to 20x, why you want to move to a higher objective and not use something which can be? And also when you add DPX and it dries with the cover slip on top of it, you can store it for as long as you want. But if you want to put oil, after a certain time there is no DPX and there is no, I mean, it's kind of a resin, by the way, DPX, which will kind of protect the section and keep it stable at room temperature conditions. So oil is only used even if you have a DPX mounted slide. When you're using the same slide for 100x or 40x, you need to add oil just to adjust the refractive index according to the objective of the microscope.
Yes, I understand.
Anything else? Nobody else? Nobody has any question? Okay, Jean, then we call it a day. Jean, Vinod, anybody?
Yeah. I think so.
We
You're done with the explanation, no? Right.
Okay. We then call it a day. Okay. Okay. See you all. Good luck for your exams.
Yeah. Thanks.
Okay.
Thanks, Salman, for joining us. Okay.
Okay.
See you.
Thank you all.
Bye.
See you.
Sir, has the attendance been taken? So can we leave?
Yeah. You can leave. No. So they will.
Yeah, you can.
Thank you.
Yeah.